Hello and welcome back. It's Marc Tennant here, and I'm talking on the third in my series on the basics of cells. Today I want to talk about cell division and a little bit more of the detail of how proteins are made. Now clearly cells need to divide. Remember, at the very beginning of life we started out as a sperm and an egg that merged to produce a single cell. And today, sitting there listening to this, you are billions and billions of cells. So we need cells to be able to divide and to continue to divide. The most common type of cell division is called mitosis. That happens throughout your body all of the time. And then there's a very rare case called meiosis. And meiosis happens really to produce sperm and to produce eggs. And we're not going to talk about that. I would recommend that you had a quick look on the internet, probably at Wikipedia or something, just to give you a little bit of information about how meiosis is different from mitosis. But our focus today will be mitosis because that's 99.999% of all cell division that happens. So we talk about a cell cycle. A cell cycle is a period through which a cell lives its life. Now this is a very abnormal sort of diagrammatic representation because it doesn't give you enough emphasis on the fact that cells can sit in this normal cell state, the green area on this graph. They can sit in this cell state for decades. Well, neurons, for example, can sit in this state for 9 decades. They go through cell division before birth and then after birth never go through this cell division process again. So the vast majority of cells in you today sit in the green area here of normal cell division. And then you go from blue to yellow to pink, and it's during that period really only occurs over a relatively short period of time in a cell's life, maybe 24 hours or so. And to give you some idea, in the blue phase here, clearly we need, if you are going to divide a cell in half, the one item in a cell that you need a perfect duplicate of on both sides, on both daughters, is the nucleus. So in the S phase, or the synthesis phase, this is the period in which the DNA is duplicated. A matching pair to the original nucleus is constructed. And this period can take about 8 hours or so to happen. After that, there is a period of rest. And then the pink phase, where we have 4 subphases called the prophase, metaphase, anaphase and telophase, is the phase that everyone focuses on. But in reality, it's a relatively short, could be an hour period in the life of a cell. The reality is this green normal cell state can be enormous lengths of times. The synthesis state, or the blue one here, is a period of about 8 hours where we duplicate the nucleus. There's a short resting period, and then we go through this very rapid pink phase with 4 subphases that sees one cell become 2 daughter cells, and those 2 daughter cells have duplicate nuclei in them. So the DNA in the cell is the critical thing here that we need to duplicate and then distribute to a cell. You would have known of the DNA because you will have heard of chromosomes. It's actually very rare to see chromosomes look as we see them in this picture, because most of the time they are spent unravelled like this, like the elastic in the middle of a golf ball, And it's only during this phase of separation of two cells during cell division that you see chromosomes as we know them normally when we see them in pictures. So let's go through these processes. Let's go through these subparts of the pink phase. So we're going to talk about prophase, metaphase, anaphase and telophase. And I'm just going to say it again to remind you, this is a very small period in the life of a cell, but it's such an obvious thing when you look at a cell that we focus on it. So the prophase. The prophase is when the duplicated DNA starts to form up and you see the chromosomes. So this is where that elastic band untangles itself and clusters up to form chromosomes. At the same time, remember I said in a previous video that a nucleus is normally surrounded by a membrane. That membrane is destroyed and the chromosomes are held in place by some of the cytoskeleton that I talked about, filaments, around where the nucleus used to be. At the same time, a little intracellular component called a centriole forms, two of them, one to one side of the cell and one to the other. And these centrioles are really anchor points where the spindles that attach to each of the chromosomes, spindles being another type of cytoskeletal component, where they attach to the chromosomes, and the centriole is going to be the anchor point where when the chromosomes split by their duplicate, one will go towards one centriole and one will go towards the other. So the prophase is this early organisational phase. The next phase is called the metaphase, and in this phase all the chromosomes line up down the centre of the cell, we're still one cell, and the spindles are connected to those centrioles. This is the lining up in preparation for splitting apart. And then in anaphase, we see the chromosomes split apart and be dragged by the spindles towards the centriole at each pole of the cell. And then finally in the telophase, we see a new nuclear membrane form. The chromosomes untangle themselves to become back to being that long string of elastic inside a golf ball, and the cell membranes starting to separate apart to split two cells into one. And here we have what it looks like in a real cell. You can see the four phases here. The prophase, when the early chromosomes are setting up. The metaphase, when they all line up down the middle. The anaphase, when the chromosomes are split in half and pulled towards the poles of the cell. And the telophase, where the chromosomes are disaggregating and the cells are separating into two. Don't forget, before this even started, the chromosome or the DNA was duplicated. So you don't forget that blue phase in that circle of the synthetic phase. Very important to keep that in mind. So that's where I want to leave cell division and spend a wee bit of time just reminding us of that process of producing proteins that I talked about earlier and add a little more to the story. What I didn't talk about before is what a protein actually is. A protein is a sequence of amino acids. There's a collection of amino acids, and it is the sequence of these amino acids that, when they're linked together, makes a protein have different functions. So depending on the sequence of amino acids and the folding that that causes in a molecule, that will determine what that protein will do. So at its heart, A protein is just a chain of amino acids linked together in a precise order. So a ribosome, as I said before, makes a protein, but how it does that is by linking the amino acids end on end to produce the chain. So you can then work backwards and go that the messenger RNA is responsible for articulating to the ribosome the specific order in which you want the amino acids to be aligned. And then you go back even further and you say that the DNA itself, all the DNA does is provide you with a blueprint of the order in which amino acids should be laid down at the end of the day. So to do that in the forward direction, we are saying that the DNA is a sequence that you can decode to then produce a protein with a specific order of amino acids in it. And that's all that DNA is. If you look to the left here in the yellow area, you can see the decoding of a little piece of the DNA. And what you can see is it's just a sequence of letters, A, T, G and C in different orders. Now it's much more complicated than that. Well, that is all that DNA is a sequence of A, T, G and C, but how it's bound together and how it's decoded and things is quite a complex process and beyond the scope of what we're going to talk about. But let's just understand that each triplet, so you take the 3 letters in a row in DNA, for example, you might take A, C, A, and that triplet will determine a particular amino acid that needs to be added to the chain. And the little table on the right-hand side here, if you look through it and find ACA, you will see that ACA represents an amino acid labelled as THR. And that amino acid would be added to the protein chain when the ribosome came across the code ACA. Now there's one little complication, a wrinkle in the story. If you notice, the DNA, I said, could be made up of 4 letters, A, T, G and C. The copy or the messenger that is made of that DNA for transporting out to the ribosome is messenger RNA. And if you look to the other side at the bottom there, you can see that the T is replaced by a U. That's just because of the nature of the chemical interactions. But all you need to do is when you read mRNA, you read the triplets with a U instead of a T. So for example, if I was looking for GCT, if I found that in the DNA, I would convert that to GCU and then look up the table there on the right-hand side, look for the GCU, and I'd see that stands for amino acid ALA or aniline. And so as that piece of the messenger RNA is read, it would then, by the ribosome, would then add an aniline to the chain of amino acids that is produced. And that's how this process works. And here you can see a diagrammatic representation of a ribosome reading off the messenger RNA and linking in a new amino acid into the chain to build a protein. I've not talked about tRNA, that's just the process that happens within a ribosome to assist it in producing this chain. You can look that up separately if you get very interested in this, but I think the basics of understanding DNA to messenger RNA, and then from messenger RNA via a ribosome to produce amino acid chain is the core to this story. So that's where I'm going to stop talking about cells for now. We've talked about cells in terms of their size and their shape and how they're grouped together. We've talked about their basic structure. What are the intracellular organelles? Mitochondria, rough endoplasmic reticulum, ribosomes, nuclei, vesicles, skeletal content. We've talked about cell membrane and how that works and what it's made of and its role. And then we've gone on and talked about cell division and those 4 subphases of cell division. And lastly, we've got the complete story of how proteins are synthesized in cells. So that's where I'm going to leave it, talking about cells for now. Please remember that as you look down at your hand, you are looking at this happening in real time in you. This is not some theoretical piece of biology. This is live and active in you today. Thank you.